Welcome to The Explainer. Have you ever looked in the mirror and thought, I have my mom's eyes, but I definitely have my dad's laugh? That question, how traits are passed down, it's one of the biggest puzzles out there. Today, we're going to trace the incredible journey from some really wild ancient guesses all the way to the precise science that finally unlocked the code of heredity. Let's get into it. That very question is what genetics is all about. For literally thousands of years, way before we had any idea about DNA or microscopes, philosophers and scientists were trying to solve this puzzle. They were desperate to understand the invisible instructions that make us who we are. So before we jump into the science we know today, I think it's fascinating to look back at some of the first ideas. These theories really show you how brilliant people tried to make sense of the world with the tools and the knowledge that they had at the time. When you look at this timeline, the sheer range of ideas is just wow. Back in the 1720s, you had the pre-formation theory. People literally thought a tiny pre-formed human, called a homunculus, was just sitting there inside an egg or a sperm, waiting to grow. Wild, right? Then, fast forward to the 1860s, and even Charles Darwin had a go at it. He proposed something called pangenesis, this idea that invisible particles called gemmules from all over your body would gather in the sex organs to be passed on. And then came a huge breakthrough. In the late 1800s, a scientist named August Weismann proposed his germplasm theory, and it totally changed the game. He argued that our bodies have two kinds of cells: your regular body cells, the somatoplasm, and your reproductive cells, the germplasm. And here's the crucial part. Only changes to the germplasm can be inherited. So if you go to the gym and get huge muscles, sorry, but your kids don't just inherit them. This idea flew right in the face of theories like Lamarck's and really paved the way for modern genetics. This whole concept of a separate, heritable material was the perfect setup for our next hero, a quiet monk whose work in a monastery garden was about to change biology forever. And here he is. Meet Gregor Mendel, the man we now call the father of genetics. He wasn't some famous scientist at a big university. He was an Augustinian friar. But what made him so special was his unique mix of skills. He was deeply curious, incredibly patient and methodical, and, this is the key, he had a really strong background in mathematics, which was practically unheard of in biology at the time. And boy, did he need that methodical mind. Just get a load of this number. Between 1856 and 1863, Mendel grew and tested almost 28,000 pea plants. This wasn't just a side hobby. It was a massive 7-year-long experiment that required an unbelievable amount of patience and precision. So you gotta ask, how did he succeed where so many others had failed? Well, it all came down to a brilliant experimental design. He chose the perfect subject, the garden pea. It grew fast, and it had these really simple either-or traits. He could also control their breeding, sometimes with just a little paintbrush. But his true genius, the real game changer, was that he applied math to biology. He didn't just look and describe what he saw, he counted everything. He analyzed the ratios, and that's how he found the hidden patterns. And this shows you exactly what he focused on, these 7 clear, distinct traits. Things like seed shape, was it round or wrinkled? Flower color, purple or white? Stem height, tall or dwarf? By zeroing in on these simple, binary characteristics, he could figure out the rules of inheritance without getting totally lost in the complexity. So from all those thousands of crosses and all that meticulous counting, Mendel started to see these predictable patterns emerge. He used these patterns to come up with principles so fundamental, we now call them Mendel's laws. They are, quite literally, the foundation of genetics. Okay, so Mendel proposed that for every trait, an organism gets 2 factors, one from each parent. Today, we call those factors genes. And the different versions of a gene, like, say, the version for purple flowers versus the version for white flowers, we call those alleles. And this brings us to another super important distinction. The genotype is the actual genetic code an organism has, like the letters TT in a pea plant. The phenotype is the physical trait you can actually see, like the plant being tall. So think of it as the difference between the architect's blueprint and the actual building. All right, this leads us to his first big rule, the law of segregation. Let's walk through it. He started with what's called the pea generation, crossing a purebred tall plant with a purebred dwarf plant. Now, in the next generation, the F1, every single one of the offspring was tall. The dwarf trait just seemed to have vanished. But then, and this is the cool part, he crossed those F1 plants with each other. And guess what? In the F2 generation, the dwarf trait popped right back up. This proved that the dwarf trait was just hidden or recessive, but it wasn't gone for good. And the math was what really nailed it. In that second generation, he always consistently found a 3-to-1 ratio of tall plants to dwarf plants. And if you look at the table, you can see how this physical ratio is explained by the underlying 1-to-2-to-1 genotypic ratio. This predictable pattern was his proof. It meant that the two alleles for a trait have to separate or segregate from each other when reproductive cells are made. But Mendel didn't stop there. He started wondering what happens if you track two traits at the same time, like seed shape and seed color. This is called a dihybrid cross, and that big grid you see is a Punnett square. It's a tool to predict the outcomes. What he found was that the inheritance of seed shape had absolutely no effect on the inheritance of seed color. They were totally independent. This discovery led to his second law, the law of independent assortment, and it explains this famous 9-to-3 to 3-to-1 ratio of outcomes. So Mendel's laws were elegant, they were powerful, but his work was basically ignored for over 30 years. It's crazy. When it was finally rediscovered around the year 1900, it kicked off the entire field of modern genetics. But as scientists started looking closer, they realized that inheritance wasn't always so black and white. There were some really fascinating plot twists to Mendel's rules. So let's check out a couple of these plot twists. You see, in Mendel's peas, one allele was always completely dominant over the other, but that's not always how it works. With incomplete dominance, the traits actually blend together, kind of like mixing paint. A red and a white flower make a pink one. Then there's codominance, where both alleles show up fully and separately, like an AB blood type, where you have both A and B markers at the same time. The snapdragon flower is a classic example of this. You cross a red one with a white one, and boom, all the offspring are pink. And what's really cool is when you cross two of those pink flowers, you get a perfect 1-to-2-to-1 ratio of red to pink to white flowers. So in this case, the physical ratio perfectly matches the genetic ratio, which is different from Mendel's 3-to-1. Human ABO blood type is just a fantastic example because it shows two of these non-Mendelian ideas at once. First, the A and B alleles are codominant. If you get both, you don't get some blend. Your blood type is AB. And second, it shows multiple alleles. For this one gene, there aren't just two options. In the human population, there are three: A, B, and O. And finally, think about this. A lot of traits aren't simple either-or categories like Mendel's peas were. Think about human height or skin color or eye color. We aren't just tall or short, right? There's a whole continuous spectrum. This is called polygenic inheritance, and it's when multiple genes all work together in an additive way to create the final physical trait. So, in just a few decades, we leaped from thinking about mysterious forces to Weissmann's germplasm to Mendel's predictable math, and then into the beautiful, messy complexities of non-Mendelian genetics. Every single discovery built on the one before it, painting an ever clearer picture of the code that connects every living thing on this planet. And you know what? The story is far from over. Right now, scientists are digging into epigenetics, complex gene interactions, and the vast networks inside our DNA. It really leaves us with a final thought. After all these centuries of discovery, what fundamental rules of our own biological code are still hiding, just waiting to be unlocked?